Go ahead. We are live, madam. You can go ahead.
Okay, okay. How many students have logged in? No one, huh?
Other madam, there.
Okay, okay. Okay, I think this will be a repetition for you. Good evening, children. And today we will be dealing with antigen antibody reactions and complement. So we have already dealt with what is an antigen and what is an antibody. An antigen is any foreign substance which enters the human body, and the antibody is the response of the immune system against the antigen, which could be a microbe or a microorganism or a foreign particle, any antigen which can stimulate the immune system to produce antibody. And these antigen and antibody reactions are useful in many ways. For example, they are used in many ways, especially in diagnosis. The main component of this antigen antibody is they combine with each other specifically and in an observable manner. These two are the salient features of an antigen antibody reaction. Specific to the antigen, the antibody will be produced and it will be in an observable manner. Why observable manner is it should be visualized by naked eye, which can be used especially in the laboratory for diagnostic purposes. And this, as I said, antigen antibody reactions, they serve several purposes and they form the basis of antibody mediated immunity, hypersensitivity, and many autoimmune diseases. And diagnosis of infection is mainly based upon this antigen antibody reactions. And that is what is in general called as serological test, which is done for various organisms and immunity markers, etc. And also for epidemiological survey. By now you must have heard for SARS-CoV-2, this ICMR is conducting epidemiological surveys where they detect the specific antibodies existing in individuals against SARS-CoV-2, which indicates immunity to the population. So it is also useful in epidemiological survey. So these are the uses of an antigen antibody reaction. And this antigen antibody reaction, it occurs only in the presence of electrolytes at a suitable temperature and pH. So these three are the prerequisite conditions for an antigen antibody reaction to take place. And there are two types of antigen antibody reactions, namely agglutination and precipitation. The difference between agglutination and precipitation is basically the nature of the antigen. If the nature of the antigen is a soluble antigen, it is called as precipitation. And if it is a particulate antigen, it is called as an agglutination reaction. And precipitation reaction is highly sensitive in detecting the antigens, whereas agglutination reactions are highly sensitive in detecting antibodies. And when will this antigen antibody reaction be visualized to the naked eye for in an observable manner, especially which is useful for the diagnostic purposes? It does not happen anywhere. It is mainly based upon a hypothesis which is called as the lattice hypothesis, where usually the antigens are multivalent, but they can bind only to the bivalent antibodies, because the antibodies have only two antigen binding sites. And during the course of illness, what happens is there is a zone of antibody excess which is called as the prozone phenomenon. This prozone phenomenon is a classical example which occurs especially in brucellosis. So how do you overcome excuse me, how do you overcome this prozone phenomenon? This prozone phenomenon is mainly overcome after dilution of the patient sample. You dilute it so that the antibody excess is reduced, so that you have a zone of equivalence where there will be equal number of antibodies and equal number of antigens. Only then there will be a precipitation or an agglutination which can take place. Whereas the later half, you have the zone of antigen excess, which is called as the post zone phenomenon, where also the lattice will not be formed. So it will not be in an observable manner. So here if you see, there are equal number of antigens and equal number of antibodies. So it forms a lace-like structure or a lattice which is visualized in an observable manner. Then coming to the examples of precipitation reaction. So precipitation reaction, as I told, is mainly it uses the soluble antigen, and it is utilized for detection of mainly antigens. And there are various types and examples of precipitation reaction. They are ring test, where you add a solution of the antigen and antibody. At the junction of the solution, you look for a ring. When this is classical example is Ascoli's thermoprecipitation test and Lancefield classification of streptococci. Lancefield classification streptococci, it is actually classified into 21 different types from A to G. So based upon the carbohydrate antigen of the streptococci, this test is done. And the slide test, the classical example is VDRL, which is Venereal Disease Research Laboratory, which is done for diagnosis of syphilis. The same way, the precipitation reaction can occur in a tube, and this is exemplified by Kahn's test, which is also done for syphilis, and standardization of toxin. This standardization of toxin is usually used in the field of vaccines. And then immunodiffusion. Immunodiffusion means the immune system, that is the immune cells or the immune structures like the antigen or antibody, can diffuse into a medium. This diffusion is mainly enhanced by using a semi-solid agar, so that because of the semi-solidarity, it will have spaces in between through which the antigen or antibody can easily pass through. One is the radial immunodiffusion, counter immunoelectrophoresis, and rocket electrophoresis. So this is the example of rocket electrophoresis, where this is the medium or the semi-solid agar, which will be incorporated with antibodies, and these are templates or holes which will be made with the template, and into this hole you add the antigen or the substance which containing the antigen. So it could be a serum, plasma, blood, or whatever it is, and then you allow it to react. What happens is the antigen will diffuse from the well into the semi-solid agar, and at the zone of equivalence it will form a precipitin line. This precipitin line is usually visualized because it is containing a dye which will make the precipitation visually to the eye. So what is this standards? There are already, the kit will contain standards which are run based upon the amount of antigen. So now you calculate the height of the rocket from the center of the well and compare it with the standard, and based upon that you can give the amount of antigen present in that particular sample. That is rocket electrophoresis. This is radial immunodiffusion, the same principle. Instead, it is diffusing radially. From the center of the well, it is diffusing radially. Here, as you measure in rocket electrophoresis the height, here you measure the diameter or the radius from the center to the outer line of the precipitation. And based upon that, you have standards, and comparing that, it gives you the approximate amount of antigen present in the sample. This is radial immunodiffusion. Then coming to agglutination reactions, as I said, agglutination reactions are highly sensitive for detection of antibodies, and the nature of the antigen is a particulate antigen. The two classical examples are slide agglutination, which is classically done for blood grouping and cross-matching in a blood bank, and tube agglutination, that is Vidal, which is done for typhoid fever, and serology, which is done for brucellosis. And there is another group of agglutination reactions, is called as heterophilic agglutination reactions. What does this heterophilic agglutination reaction means is, because of sharing of antigens, that property is utilized in this test. For example, wheel Felix test is done for typhus fever, which is a rickettsial disease. It is very difficult to cultivate rickettsia. So getting the antigen of rickettsia is very difficult. So what they have identified is this rickettsia has antigens which are similar to that present on Proteus vulgaris. So due to this sharing of antigens, Proteus vulgaris is used as an antigen, and the antibody in the patient serum is detected against it. The same way Streptococcus mg and cold agglutination test, which is done for primary atypical pneumonia, which is mainly caused by Mycoplasma pneumoniae. Then you have the Coombs test. Coombs test is a main test which mainly detects incomplete antibodies. A classical example of incomplete antibodies is the Rh antibodies, which occurs in erythroblastosis fetalis, which occurs when the mother is Rh negative and the father is Rh positive and the fetus is Rh positive. So then you have the passive agglutination test, where this property of the antigen, conversion of a soluble antigen to a particulate antigen by coating on it with latex, RBCs, beads, etc., is called as the passive agglutination test. This is the Coombs test, as I told you, it detects the incomplete antibodies. There are two types of Coombs test, direct and indirect. Direct Coombs test, what happens is the sensitization of the RBC occurs in vivo or within the human system, that is in the mother's environment. So this sensitized RBCs, which contains the antibody, it will not agglutinate because it is an incomplete antibody. So what we do is we take the mother's sample and we add antibody to this antibody, that is anti-human IgG antibody, which is called as the Coombs reagent. This will now agglutinate this non-agglutinating antibody and produce agglutination. This is called as the direct Coombs test, where the sensitization occurs within the or in vivo. Whereas indirect Coombs test usually occurs in vitro or in the laboratory. The classical example, a mother who is Rh negative can have prenatal testing to determine the presence of non-agglutinating antibodies in a bloodstream, which will attack the fetus RBCs if the father is Rh positive. So prenatally, you take the patient sample and sensitize it with the incomplete antibodies, and then you add the anti-human immunoglobulin. So this will cause agglutination, and this is called as the indirect Coombs test. That is about the Coombs test, which detects incomplete antibodies. And the other agglutination reactions are the complement fixation test, which is classically exemplified by Wassermann's test, which is also used for diagnosis of syphilis, and neutralization test, which is used for neutralization, identifying the presence of viruses and toxins. Apart from that, immunofluorescence. What is fluorescence? Fluorescence is a property of a dye which absorbs light of one wavelength and emits light of a different wavelength. So this emitting light can be visualized using a fluorescent microscope. So what you need is, you need a fluorescent labeled antibody, and the fluorescent dyes which are commonly used are auramine, rhodamine and fluorescein isocyanate. The same way we have radioimmunoassay also, where instead of the fluorescent dye, you can tag an radioactive isotope like iodine 131, etc. And you have to look for the radioactive emission by means of a Geiger-Müller counter. Hey. So this complement fixation test, it is usually conducted by means of adding an indicator system. So what is this indicator system? It is the RBC which is sensitized with hemolysin. So what the main function of this complement is lysis. The main function of complement is it causes lysis of the cell to which it is attached. And complement, it cannot attach directly to the cells. It has to attach only with an antigen antibody complex. So when an antigen antibody complex is formed, the complement will be utilized. So here if you see, you are adding a serum which contains the antibody and then the antigen. So the antigen antibody reaction takes place, and this is the antigen antibody complex. So now when you add complement, the complement will be utilized or attached to this antigen antibody complex. Later, when you add the sensitized RBCs, the complement which is already attached to this antigen antibody complex will not be available to lyse the RBCs. So the RBCs will not lyse, and this indicates that the reaction is reactive or the sample contains antibodies. The other way, if the antibodies are not present in the serum, you add the antigen, there won't be antigen antibody complex. And when you add the complement will be free existing in the solution. Now when you add the indicator system, the complement which is free in the solution will attack the RBCs and cause lysis. So lysis of the RBCs indicates the absence of antibodies in the sample, and no lysis indicates the presence of antibodies in the sample. This is complement fixation test. Then coming to ELISA, you know ELISA is enzyme-linked immunosorbent assay. It can be used for both detection of antigen or antibody. If you want to detect an antibody, the antigen will be coated in the well. If you want to detect the antigens, the antibody will be coated in the well. And this test, it consists of a series of washing procedures to remove the unbound material. So this is the antigen which is bound to the well. And now if you add the sample which contains the antibody will attach. So next step is washing to remove this free antibody. So this bound antibody will be now present. And to this bound antibody, you are adding a conjugate. This conjugate is nothing but a combination of an antibody to this antibody which is tagged with an enzyme. Once again, you do a washing procedure to remove the excess of the antibody conjugate, and later you add a substrate. So once when you add the substrate, the enzyme which is attached to this antibody will release, and it will act upon the substrate and will produce a color change. This is how you detect the color change by means of an optical density, and each kit gives a calculation based upon the positive control and negative control, and it gives you whether the sample contains an antibody or an antigen. You can also detect the antigen, where the antibody has to be coated inside. Now ELISA is used for most of the diagnostic investigations in the laboratory. Then you have the blotting techniques. Blotting technique is nothing but you individualize the cells into either DNA, RNA or proteins based upon the molecular weight, and you blot them into a nitrocellulose membrane or into a gel, and then you look for the antigen antibody reaction. So if DNA is used in the blotting technique, it is called as southern blot. RNA in northern blot, and proteins, it is called as the western blot. Western blot, you know very well, is a confirmatory test which is done for diagnosis of HIV, where you can see the individual proteins which are separated onto the nitrocellulose membrane, and you can see the band. This is the standard band, and see here you can see the GP160, GP55 and P24. So this indicates it is positive. This is only P24 antigen. It is usually done for diagnosis of pediatric HIV patients. So this is how you diagnose using a Western blot technique. So coming to the questions, which of the following is included in the indicator system of a complement fixation test? Specific antibody and complement, specific antigen and complement, red blood cells and hemolysin, guinea pig serum. Anyone?
Third option.
Pardon?
Red blood cell hemolysin.
Red blood cells is the hemolysin is the indicator which is commonly used in the complement fixation test. And no lysis indicates a positive reaction, and lysis of RBCs indicates a negative test. Okay. Then which is not a heterophilic agglutination test? Wheel Felix, Vidal, Falbanel, Streptococcus mg.
Vidal test.
Vidal test is done for diagnosis of typhoid fever. Okay, it is mainly based upon the antigen sharing between the organisms. Prozone phenomenon is a feature of which disease?
Brucellosis.
Brucellosis.
So to overcome this prozone phenomenon, you have to do serial dilutions of the serum and then do the test. Okay, serial dilutions to do the test. The following methods of diagnosis utilize labeled antibodies except ELISA, heme agglutination inhibition, radioimmunoassay, immunofluorescence.
Option B, heme agglutination.
Because ELISA uses enzyme as a labeled antibody, radioimmunoassay uses antibody tagged with radioactive isotope, and immunofluorescence, it uses the fluorescent dyes. In the direct immunofluorescence for identification of a specific infectious agent, fluorescin may be conjugated to the microorganism, the sheep red blood cells, antibody specific to human gamma globulin, antibody specific to microorganism.
C, ma'am.
There are two types of immunofluorescence, direct and indirect. So antibody specific to the microorganism. During direct immunofluorescence, the fluorescent will be directly attached to the antibody, which will react with the specific pathogen. So just it is a two-step procedure. Whereas from indirect immunofluorescence, you have to add an anti-immunoglobulin against the antibody, which will be tagged with the fluorescent. Is it clear? That is the difference between direct and indirect. Direct is a two-step procedure. Indirect will be a three-step procedure. The radioimmunosorbent test is a technique used to measure total IgE concentration. As we detect IgM, IgG, and then IgA, etc., IgE is very difficult to detect in precipitation and agglutination reaction, for which you need a specialized test which is called as the radioimmunosorbent assay, which is usually used for detection of the total IgE concentration. So this specific IgE levels are measured also by radioallergosorbent assay, which will be specific for the specific allergen. A few allergens which I can name is pollen grains, house dust, etc. Even this cat's hair, dog's hair, all these can be allergens. So specific to the allergen also you can do the test, or in general the total IgE can be detected by means of radioimmunosorbent assay. Which of the following can be used in passive agglutination test? What is passive agglutination test? Conversion of a precipitation reaction to an agglutination reaction is called as a passive agglutination test, which alters the soluble antigen to a particulate antigen. So these soluble antigens can be attached to either a latex particle, RBC, or carbon particles. So all these can be used in the passive agglutination test. Okay, so converting precipitation to agglutination, it is mainly adsorbed to a carrier particle. Okay, a professor gives a student a preparation of a fluid antigen and asks the students to arrange serological tests to determine the presence of antibody in this antigen. To this antigen, of the following procedure, which would be the most sensitive and suitable test? Which will be the most suitable test? Anyone?
Option D.
Okay, passive. See, the antigen is fluid. So it is a soluble antigen and you want to detect the antibody. If the antigen is soluble,
Okay, ma'am.
what is the reaction it will take place? It is
Precipitation.
precipitation. But here you want to, and precipitation reactions can detect only mostly antigens. Agglutination is the test which is useful for picking up an antibody. So here they have asked to detect the presence of antibody to this antigen. So you have to convert this antigen to a particulate antigen and then do an agglutination reaction to identify the specific antibody to this antigen. Is it clear? Okay.
Yes, ma'am.
Okay.
Okay, ma'am.
So that is it. Allergosorbent, which I have already told you. So with that, we finish the antigen antibody reactions and coming to the complement. So what is this complement? Complement is a part of innate immune system. Innate means which will be present since birth in an individual. And it is a protein present in the serum which provides non-specific innate immunity. It is not specific. Always immunity we say it is specific, because even immunological reactions we say it is specific, but this complement is a non-specific innate immunity. And complement will be present in sera of all mammals, and non-specific serological, it is a non-specific serological reagent. It is heat labile, and complement, it does not bind to free antigen or antibody. It will combine to or bind to only an antigen antibody complex. All classes of antibody does not bind to the complement, because this complement, it usually binds to the CH3 domain of the antibody. You know, there are various domains in the heavy chain of the antibody. Of this, the CH3 domain of the antibody is the attachment site for the complement. Only IgM, IgG3, 1 and 2 will fix the complement in order of preference, whereas IgG4, A, D and E will not fix the complement. Is it clear? These are a few MCQs which they commonly ask. So what are the components of complement? They are represented by the capital letter C, and they have 9 different fractions called as C1 to C9. C1 as such has 3 subunits, C1, Q, R and S. C1 to C9 are named in a sequence of reaction like C1, C2, C3, C4 to C9, but with a change of order, whereas after C1, C4 comes, after which only C2 comes, then C3, C5, C6, 7, 8 and 9. Okay. And complement is inactive and it will get activated only in the presence of an antigen antibody complex, where the complement will bind to the CH, and complement components cleaves into dissimilar components. That is, when they are activated, they cleave or divide into 2 dissimilar components. The larger fragments usually joins the cascade, whereas the smaller component, which is usually given the name A, is released into the circulation, which has biological effects like anaphylatoxin, vasodilatation, vasoconstriction. It can cause many biological effects, which is the active part of the complement components. Okay, these are in general about the components of the complement. And this complement can be triggered off by 2 parallel but independent mechanism of pathways, which differs only in the initial steps, after which they will have the similar steps from C3 onwards. Okay, they are the classical and alternate pathways. What is this classical pathway? Classical pathway is usually stimulated by only immunological means, like an immune complex or a DNA or a C-reactive protein. Whereas alternate pathways can be activated by non-immunological substances like zymogen, bacterial endotoxin, IgA or IgD. Is it clear? So classical needs only an immune complex, whereas alternate does not need an immune complex or immunological means for activation. So this is the classical pathway where you can see you need an immunological reaction. So an antigen antibody complex. This C1 binds to the C3 component of the antibody and it cleaves into C1A and C1B. So this C1A which gets activated will act upon C4, which is because after C1 in the cascade you have C4. This C4 will cleave into two dissimilar fragments, C4B which is the larger fragment and C4A which is the smaller fragment. This C4A has anaphylactic activity whereas C4B will join the cascade. This C4B will now act upon C2 and it will release C2A and C2B. So this combination of C4B and C2A together is called as the C3 convertase because this will act upon C3 to cleave C3 into C3A and C3B. So this is called as C3 convertase. So this is the common junction where the classical pathway and alternate pathway differs. Here it is the antigen antibody complex which cleaves C1 to C1A. This acts upon C4 to cleave it into C4A and C4B. C4B will join the cascade, act upon C2 and it will release C2A, and so this will be C4B and C2A will form the C3 convertase. So this C3 convertase will now act upon C3 to release C3A and join C3B in the cascade. The same way here, any a biomaterial surface like zymogen, etc., they can stimulate C3. There will not be action of C1 or C2, C4 here. They will activate C3 and the C3A will be released and C3B. Here you have a factor D which will come into action. This, in the presence of factor D, it will act upon a factor B which is also present in the serum, and this factor B will divide into two dissimilar fragments, capital B and capital BA. So this capital B A will have anaphylactic activity, whereas C3B and this factor B is called as the C3 convertase. This will also now act upon C3 and cleave it into C3A and C3B. So this C4B, 2A and 3B is called as C5 convertase, which will act upon C5. The same way here, C3B, BB and 3B will act upon C5 and that is called as the C5 convertase. This C5 is cleaved into a larger fragment and a smaller fragment C5A, and this larger fragment C5A will now join the cascade where it will be fused with C6, C7, C8 and C9. So this C5B, 6, 7, 8 and 9 together is called as the MAC complex or membrane attack complex. So what is the main function of complement I told you? It is lysis of the cell. So this membrane attack complex will attack the cell membrane of the targeted cell and it will form numerous holes so that it extrudes all the cytoplasm and the internal contents, thereby causing lysis of the targeted cell. That is the main function of this complement. Is it clear?
Yes, ma'am.
Any doubts regarding this? Shall we move on? Okay. So this is what the classical pathway, antigen antibody complex, C1, C4 and C2. All these together will act upon C3. Here alternate complement pathway in the presence of factor D and factor B will act upon the C3 and it will release C3A and C3B. C3A, it stimulates histamine release, increased blood vessel permeation. All the anaphylactic actions are mainly by C3A, whereas C3B will join the complex, which will act upon C5 and release C5A, which also is reason for this inflammation. And then the MAC complex, which is fusion of C6, 7, 8, and 9. So this will form numerous holes, the membrane attack complex, and it will cause target cells. It will release all the cytoplasm and contents within, and finally cause lysis of the cell. Okay, so that is the classical and the alternate complement pathway. Then each component of this complement is synthesized in the human system at various levels. C1 is mainly biosynthesized in the intestinal epithelium, whereas C2 and C4 by the macrophages, C5 and C8 by the spleen, and C3, 6, and 9 by the liver. Okay, so these numbers where it is biosynthesized, you have to remember because they may be asked in the MCQ. Then, what are the biological effects produced by this complement? It mediates lysis, promotes phagocytosis, immune adherence, and opsonization. You know, opsonization is engulfing by the phagocyte of the targeted microorganism. And complement also participates in type 2 and type 3 hypersensitivity. What is type 1 hypersensitivity mediated by? Anyone?
IgE.
IG?
IG.
Whereas complement is a major component in type 2 and type 3 hypersensitivity. And apart from that, it acts as an anaphylatoxin. And immune adherence and opsonization with the help of the C3b receptors, which is present on the phagocytic cells, where the complement may go and attach to the C3b receptors and enhance opsonization. Chemotactic effect will be mainly by C5a and C5b and 6 and 7. Anaphylatoxin by all the A, C3a, C4a, and C5b. And cytolysis is the membrane attack complex, which is responsible for lysis of the targeted cells. And excessive C3 activation may lead to endotoxic shock. So these are the few biological effects the complement can cause on activation. One specific mention about C5a, it causes neutrophil activation, increased expression of adhesion molecules, immigration of neutrophils, and chemotaxis. Chemotaxis is attracting the cells to the site, and monocyte activation, that is the lymphocyte activation, and mast cell degranulation. Mast cells are mainly involved in IgE or the type 1 hypersensitivity. And C3, they are less active than C5a. They enhance platelet adherence. They enhance opsonization and phagocytosis, as well as they act as an anaphylatoxin. These are the biological effects of C5a and C5b, which are released on activation of the complement. This is opsonization. This is the macrophages, which will contain the receptors for the complement, and you have the antibody. So this antibody antigen complex along with the complement will enhance phagocytosis, because it is the antigen antibody complex which will help in attachment of the complement, which enhances opsonization or engulfment of the antigen by the macrophages. And deficiency of these complements can also lead to certain diseases. C1 inhibitor is an inhibitor which will inhibit the activation of classical pathway. And if you have C1 inhibitor deficiency, you develop hereditary angioneuritic edema. And C1, C2, and C4 deficiency will lead to collagen vascular disorders. Deficiency of C3 may lead to recurrent pyogenic infections. And C5 to C8 may lead to bacteremia and toxoplasmosis. So these are the few deficiencies of the complement which may end up in few diseases. Now we'll move on to the questions. Which of the following complement components are required exclusively in the classical pathway? C1, C2, C4,
C4.
A.
Option A, ma'am.
Very good. Because the alternate complement pathway does not require C1, C2 and C4. Instead, they go
C3.
directly to the C3. Which of the following proteins activate neutrophils and also causes them to become sticky and adhere to the vascular endothelium? 2 you can rule out. C4B, B and C you can rule out. Is it C3A or C5A?
D.
C5A, very good.
C5A.
So it activates neutrophils and also increases the adhesion molecules, which enhances the stickiness of the cells. Okay. Classical complement pathway is activated by IgM, IgG, none, both.
Both, ma'am.
Both.
How? Can an antibody alone stimulate a complement cascade?
No, ma'am.
The antigen and antibody complex. Antigen alone and antibody alone cannot stimulate the complement pathway. So it is none. Okay. You will be usually misguided, but it is usually none.
Yes, ma'am.
Effector functions of complement include all of the following except attracting phagocytes to the site of infection, facilitating phagocytosis of complement-coated bacteria, increasing blood vessel permeability to plasma proteins, presenting antigens to B cells.
C.
C,
Ma'am, D. D.
B or D?
D.
D.
D. Yes, D is the answer because antigen presenting will not be a function of the complement. You have
Help us.
separate cells called as antigen presenting cells, which could be your phagocyte or your B cells also, or there are various Langerhans cells which are present in our system, or macrophages can also act as an antigen presenting cell. But the other three are the functions of the complement. Which of the following are least sensitive to complement mediated lysis? Enveloped viruses, erythrocytes, gram-negative bacteria, gram-positive bacteria. I'll tell you the answer. It is usually the gram-positive bacteria because all gram-negative bacteria, they possess a lipopolysaccharide, which is also called as the endotoxin. Endotoxin activates the complement. So once this gram-negative bacilli are present, the endotoxin released will activate and the targeted cells or the gram-negative bacteria will be killed. Whereas in the gram-positive cocci, the surface is rich in sialic acid, where C3 binds to factor H instead of factor B. And this factor H stimulates the catabolism of C3. So the C3 will never be activated in case of a gram-positive bacteria. Is it clear? The same way the enveloped viruses, there are certain enveloped viruses like example Epstein-Barr virus, which has a complement receptor 2. So this complement receptor 2 enhances the attachment of the complement to the cell and which leads to lysis of the cell, which occurs especially in Epstein-Barr viruses. Okay, so that is why it is the gram-positive bacteria which are least sensitive to complement mediated lysis, but all others are usually stimulated by a complement mediated lysis. Okay. In the complement system, true is alternate pathway does not rely on antibody. Classical pathway is best activated by bacterial endotoxin. C1 is the first enzyme complex in the classical pathway. The membrane attack complex is made up of C3A and C5A.
Ma'am, C.
Good. Because alternate pathway does not rely on antibody. Classical pathway is best activated by endotoxin is also not true. And the membrane attack complex, what is it made up of? C3, C5A, 6, 7, and 8 and 9 together is called as the membrane attack complex. So C is the correct answer.
7, 8,
The membrane
7, 8,
attack
9.
complex. Pardon? In the membrane attack complex phase of the classical complement pathway, the role of C5b is to activate the C5 convertase activity, attract neutrophils to lyse the pathogen, initiate formation of MAC or membrane attack complex, polymerize into membrane spanning channel. It is initiating the membrane attack complex. C5b, 6, 7, 8, and 9. That forms the membrane attack complex. So the main function of C5b is to initiate the formation of membrane attack complex. Okay.
Yes,
As
ma'am.
far as C5a is anaphylactic as well as chemotatic, which we have already seen. Is it clear?
Yes, ma'am.
Of the following are anaphylatoxin released in the complement pathway. C3a, C5a, both, none.
Both, ma'am. Both.
Both, ma'am.
Both of them. Complement participates in the following hypersensitivity reaction. Type 2, type 4, type 1, all of these.
Type 2, ma'am.
Type 2, because type 2, 1, 3 only involves the component. Type 1 mainly involves the mast cells and IgE, and type 4 it mainly involves the lymphocytes, which forms a granulomatous reaction.
Yes, ma'am.
Okay. Okay, with that we finish the class on
Compliments.
complement and antigen antibody reaction. Okay, so today you just go through this so that you remember it, and next we will be going into, I think, structure of the immune system and the immune cells, which are the most important aspect of immunity. It will be little vast, but we will see in a nutshell. Okay.
Okay,
Any
ma'am.
Yes, ma'am.
clarifications? Okay, you can read, and if you have any doubts or clarifications, you can get your number from Dr. Umesh and WhatsApp me. I can also clarify to you at any time. Okay.
Okay, ma'am.
Thank you.
Thank you, ma'am.
Thank you, everyone. Madam, shall we finish the meeting?
Yes, sir.
Thank you so much, madam. Thank you so much.
Thank you.
Thank you
Thank
so
you.
much, all.